Got it.
All right. So I'm super excited today to introduce Professor Eva Strauch from University of Georgia. Eva is, I would say, a pioneer in de novo protein design. I think she did the world's first requirement-driven protein design with her fold from loops algorithm. And so I'm always excited to see what's going on in her group. So yeah, Eva, please take it away.
Thanks. All right. It wasn't fold from loops. It was anyway, it was hotspot based design. But anyway, one of those. All right. So let's see. Because all right, here we go. I'm trying. Normally I have two screens today. I don't. Has something to do with children in the house. Okay, so I want to talk about a few things we're doing. So we're relatively new lab. So we have a few projects going and that have now data coming in. And so I thought I'll talk about the most advanced ones. Let's see. Sorry. I didn't realize it's so much different when you have only one screen. All right. So yeah, so the first thing I wanted to talk about was talking a little bit about multi-state design, which we've been using for viral surface proteins, because that's what we biologically are very much interested in. We're curious about these homing devices. Then I'm going to talk about shortly about de novo design of small folds and what platform we have for that, and where this can go, or what we extracted from it. And then we have a whole set of new design algorithms that involve neural networks for the design of protein-protein interactions. And so, yeah, all right. So each a little bit, as we now have data on all of these projects. All right, so just a short background without wanting really to go into the details of how these viral surface proteins work in the context of membrane fusion. But you can imagine you have two membranes, host and viral. They have to get together in some form. It's not very energetically favorable. So therefore, the virus has proteins that do that job. And so the energy is stored in their conformation such that they recognize the host cell membrane. Then they have a variety of different intermediates that we actually don't really have good structural information about, but lots of hypotheses. Now we're starting to get some data on it. And then eventually another conformation that is highly energetically favorable. And that's what in the end is supposedly fueling the membrane fusion. So as a biochemist, I'm like, okay, we have a start and a stop. Let's look at start and stop and ignore the rest for a moment. And I like to think about it in the context of energy. One is a higher energetic state and one is a lower, much more favorable energetic state. And there might be X amount of intermediates. And so now we have structures for both of the start and the end points. And so one thing to think about is if you think what the immune system sees, you would think, it's the pre-fusion state, because once it's post-fusion, either the damage has been done or, well, the damage has been done. So the most effective way to inhibit viruses is to target the pre-fusion state. So essentially, if you're able to stabilize this conformation over the other, you might have a good vaccine. I mean, obviously, multi-state design on different conformation is applicable for many different things. So, but this is our application. So in this context, so RSV is nice because it's been studied pretty well in context of its epitopes, and there's antibodies available. And we know that, for example, this top epitope, is pretty neutralizing if you make an antibody against it, and that it's not very recognizable in its post-fusion state. So again, coming up and thinking about this in the background, so we want to stabilize the pre-fusion part. So we went ahead and looked at, what do mutations do when you do it on either conformation, and so then we get a pretty nice difference between post versus pre-fusion, and identify relatively easy which one stabilizing which conformation well. And so the idea then is, can you take those positions that are easily affected by mutations, or in this case with allele mutations, and can you look into all possible substitutions and learn a little bit about what's stabilizing, what's destabilizing. And then he can use this and put that essentially in a simple fitness function, just to see, like, we want one over the other. And this is work done by credits to my lab, Karen Gonzalez. And so she identified the important mutations that needed to be made, had all possible point mutations, and that's what she used to then make 3 different variants. And so here's the energetic differences. And you can see, like, pre-fusion actually was improved. That was our starting point, energy. And then, while the post-fusion got a little bit improved too, the difference is relatively big. And so we tested these mutants, or variants, I have to say, and we first do cytosol chromatography to see, do we have trimers that are not aggregates, and then we compared it to DSCAF, which is the current clinical candidate, and A2, which is the original strain sequence, which immediately actually goes into its post-fusion state. It's not very stable at all if you make it on its own. So then we can take antibodies to check. We can take antibodies to check whether the pre-fusion state is actually intact. And so here, what we would expect from the original strain, which is mostly in this post-fusion, you don't get much binding, but both BSCAF and our design has binding very nicely. Then you can look into differential thermal fluorometry. And actually, well, anyway, so this is then the derivative, and you can see the melting points roughly. It's here it starts to melt, where we're a couple degrees later and melting, meaning we have comparable free energy in our system now. And then we were, of course, curious, did we destroy the trimer or not? Does it look how it's supposed to? So if you do negative stain, you should be seeing blobs, not rods. And so this is some data we got from our collaborator at the Musa Lab here on campus. And so we saw blobs, not rods. And he actually solved the structure, well, Jiachen and his students did, which is what 3.8 angstroms. And the structures actually were the model versus the crystal structure were about 1 angstrom different, which means we're pretty close to what we thought we were going to get. So then, of course, you want to know, when you put this into mice, does it behave similar to the clinical candidate? And so here in gray is the DSCAV1, originally made by designed by McLellan, and our design MutDisk1. So they compare, and ELISA data is similar, so they have the same amount of antibodies. And then you, of course, want to know, well, it's one thing to make antibodies, are they actually useful? And so the answer is yes. So it's just different dosages, and with and without adjuvant. And so, the control antibody is somewhere in the middle, and depending on whether you have a higher or lower dosage, you're a little bit better or worse in neutralizing the mouse. So short version, we made something that was comparable to that current DSCAF version, using multi-state design and only a few mutations that stabilized the rather large surface protein, and it elicited neutralizing antibodies. So that was pretty exciting, because it was a direct application on how one can use multi-state design to get for stabilization. And so the thing is, like, something works, then you want to know, well, I mean, we know actually the melting temperature. Does it still bind the post-fusion state because it's post-fusion? I mean, a multi-state design that shouldn't really allow the post-fusion state anymore, and that's exactly what happens. So we actually essentially, what we tried to do, we tried to destroy the post-fusion conformation, and so our variant, when we heat it up enough that it should be starting to like melt or pop, just enough not to completely aggregate, And so the answer is yeah, so we just destroy that conformation and stabilize the other one, which is exactly what a multi-state design should be doing. So then here is the multi-state design version 2.0, because then we want to know, we established this in context of these proteins. This protein for RSV is it applicable for other viral surface proteins? And so there's a cousin to RSV, which is metanomavirus. So actually it's been till like a month ago. It hasn't been reported that you can just make this protein in mammalian cell lines. You have to use a specific Yersinia expression system in order to get any protein out of it. So it's been incredibly hard to make this protein. So obviously there's no vaccine. There's also no therapeutic available for metanomavirus, and it's maybe not as bad as RSV in context of its mortality rates. So it's mostly affecting children and older people. But so we were interested, does our protocol work there too? Is this a good automation to stabilize viral surface proteins as for vaccine candidates? And so we had to work a little bit harder on this because it turned out we had to change protocol, look at more mutations, etc., to see that we can get the difference between the energy between pre and post fusion a bit higher, and eventually succeeded. Again, that's Karen who did all this work. And so we expressed our four candidates. Two of them look great. This is the control, which is the existing crystal structure, which you can see has a lot of aggregates, and our construct had a really nice clean trimeric peak. Also like a 10-degree improvement in its melting temperature as our measurement for the free energy in the system. And so, of course, you want to know how does it behave when you use different antibodies. And the answer is it doesn't bind. So MP8 is, binds to the site. It's pre-fusion specific. So it doesn't bind the monomer control. It doesn't bind the post-fusion, but it binds the original control as well as our design pretty well. So that's encouraging. And then we have some negative stain that we're currently trying to get a little bit better data from. But you can see it's more plumps and rods again. Again, these is preliminary data, but they're still showing we have trimeric protein. And so we actually just got the information that the crystals that Jared's lab grew diffracted at 2.4 angstrom, which is a pretty cool low resolution for these big proteins. And so hopefully soon we know how close our design was from our model. So that's coming up. Oh yeah, so this is the negative stain. And of course, since we have SARS-CoV-2 going on, we need to know, can we use it for SARS-CoV-2? So this is this enormous pre-fusion spike protein conformation in the post-fusion state here. We had to buy more memory because it just occupied a lot. And so we made a variety of designs where we stabilized the pre-fusion state over the post-fusion state and tested 5 of them. And so here's the summary. Again, we'll use melting temperature as an indicator for stabilization. And so our control started melting at 45, but our designs are around 61, which was exciting because that meant we actually stabilized quite a bit. And so then we were like, well, we didn't have stem antibodies, and RBD or not doesn't really make a difference. I think the rest melts. So we wanted to know, oh, right, well, we can test binding to its receptors. So we expressed ACE2 that we then use for bilayer interferometry to see, does it still bind? And the answer is yes. And then we looked into negative stain to see, do we have pre-fusion? And so the answer is yes. I mean, it's one of the most gratifying negative stains because it's such a big particle that it makes just pretty pictures. And so then we started collaborating with the core facility at ASU, where you can get cryos data. And so we got our first big set of pictures that we then processed ourselves. So we are getting a little bit into cryo so we can solve our structures. And so we were able to solve. Well, we haven't solved the structure yet because we're still in the process, but we were able to extract a 2.7 angstrom map out of our design, which is right now aligned to a 1-up RBD. Interestingly, there wasn't that much closed to open, all open, or 2-up RBD versions, but it was mostly just 1 RBD up. So this was exciting because we had never actually done this. We never solved the structure before. So we're in the middle of this, and I'm pretty excited that this is actually something we potentially could also add to our pipeline of making proteins work. Okay, so then now going a little bit more into the novo design and modeling for proteins, or I guess this is mostly Rosetta in this section. We were very interested in figuring out how we can make a large database of protein scaffolds that are a little bit bigger than the 42 amino acids that were previously reported, because if you have just 20 amino acids more, you actually have at least 2 secondary structure elements you can add to a protein. So that allows you actually to make more folds and more complex folds. And the idea is that, let's say, if you want to make binders, that you need to have a lot of shapes, and that's essentially the idea here. What can you make? How diverse can these small folds be? And how can you control this shape diversity? And so the folds we looked into making are listed here. So essentially 3 and 4 helical bundles, Coil's version of it, beta grasp, thyrodoxin, and you can also make a small thyrodoxin out of this. And actually also 6-plated beta propellers, but we didn't follow this up, so I can't report too much on it, but we can make them on the computer that look pretty, and they also express. And then I made up 2 folds, which are called F2 and F4 for Fantasy 2 and Fantasy 4. So the idea too here is, right, we want scaffolds for new function, and also scaffolds to be able to learn a bit more about proteins and be able to fuel some machine learning protocols to increase the overall success rate on the novo design. And so here's an example for like the new pipeline we developed. So here I'm showing only like 20 different versions of beta grasp fold where we varied something that I'm going to explain in a second, and you can see there's quite a bit of structural diversity, which is what we want in order to make, let's say, a lot of binding proteins. And so the objectives are that they need to be chip encodable, meaning like the amino acid lengths cannot succeed whatever the current chip technology allows for oligonucleotide pools, which at this point we're 230 bases, which translates to 64 amino acids, given stuff you need for cloning. And we wanted high shape diversity and data for machine learning. So, okay, I'm going to go quickly through this because people are familiar with that. But like proteins in Rosetta, either for prediction or for de novo design, are folded by using fragments coming from the PDB, right? So like fragment insertions kind of looks like this. You start seeing elements forming all over the protein because they get inserted, they get evaluated. You have the progression of Monte Carlo going on, so then after 1 million hours later, you may have your protein. I'm not going to play this through because I feel like this is everyone's familiar with that. Okay, so the idea here is that, well, originally a lot of the novo design and Rosetta, and again, this is because I thought this started as a Rosetta modeling club, so I thought this might be interesting. So originally the way you define a new fold is by defining its sequence, and there you should think about, instead of just having helix, sheet loop. You can be a little bit more specific if you divide the Arbeitsschanten plot in those 5 sections that we then call a bagel. And so you can essentially have 5-letter alphabet to describe a fold as a whole sequence. And so then you use fragment insertion to fold up the fold. See, can you make something that resembles somewhat naturally possibly looking protein because you have local fragments in there. And because you do this over and over by inserting fragments, you have an incredibly inefficient process of folding up the protein. You may have some constraints because you have hydrogen bonding between sheets, etc. But overall, it's not really well guided, and I think we can do much better than this. And so the idea, and the limiting factor is also once you have larger proteins, this does not scale well. And so the idea is, can we focus on building the proteins sequentially, finding local perfection, and thereby A, making it computationally more efficient, and B, also making sure you fulfill each step the requirements needed in order to build your fold. So this is essentially what we have incorporated on the pipeline, which we call the fold architect. This was done with Tom Linsky together, who wrote most of the core code, and then we just designed the proteins. But the idea here is that, if you want to define the fold, you want to give, of course, what kind of secondary structure elements there are, and then ranges of the secondary structure elements. But you don't really want to make definite decisions on this only needs to be this one Abego code, but you want to say I want to have 4 sheets, and there can be between 5 to 8 residues long, and then you can have a few distance constraints that are also dynamic because you don't really know the ranges. You can think about register shifts you would like to have, and of course bulges, which allow you to have curvature in sheets. So those are the main parameters we are allowed to change. And the idea there, then, folding process in silico, you take into account all the already existing folding principles, what kind of connectors are between different types of sheet helix loops, etc. And then you can filter, of course, on geometries and do sequence design. There we actually tried out a variety of protocols as well. And then you can screen it experimentally using yeast display and digestion proteases. And that would allow you to refine the principles behind de novo design for ideal protein folds. And so that's essentially what we did. And so in context of refine, we added a variety of connectors for alpha, because that wasn't really well implemented. And then the overall architecture to have a dynamic description of a fold. So what else is in the overall architecture, fold architecture, is you can define sheet triads. You can, as I said, the distance constraints, you can tell what is parallel and anti-parallel. And so, for example, if you had the sheet architect, you could define, your 4 sheets where you say 4 and 1 should be up, 1 should be down, and then you can specify the ranges. Again, this comes off one. You can actually have the loop description too. So you can use the precise Abego code, or you can have length variations as well. So each element, you have to decide where you want to start. And sometimes I have to admit that it is not that you start at 1, and sometimes you have to start in the middle. So this a little bit depends on the fold, but you can let a variety of trajectories go and see what produces the most output, and that typically works pretty well. So we use that for all of these folds that I showed earlier. And again, why is all of this possible? Why would we bother to go into to develop something where we can make thousands of designs? Because, well, we can now order thousands of designs at a time. If they're not too long, I already mentioned that, they have to be oligonucleotide pool arrays. And then the other part is yeast display, and that if you do titrations, using a few proteases, you actually can get an estimate on the overall energy or stability, I guess, if you want to translate that, of a protein, because the quicker something digests, the less stable it is, or the less folded it is. So if it's not very folded, it's quickly digested. And so it turns out that actually correlates with its free energy. So those are the two tools we can now use. So another example, again, remember how I told you that, a lot of de novo design has been done with blueprints where you just have one blueprint for one fold, so not much structural diversity, but now using this dynamic way of describing a fold. So if you actually then, made a few backbone designs and then look at what is the ideal combination, you actually get quite some diversity. And each one would be potentially like the one single blueprint, but you don't have to write the blueprints anymore. And also, again, there underneath the hood are the hydrogen bonding sheets, the definitions about the distance constraints, let's say like sheet to helix, and let's say angles of sheets and helices, etc. So all of these are there, and they can help guide the search space, which makes it much more efficient to actually make these geometries. So here's an example for these different folds, and so if you just let them run, what kind of length distributions they would have. Obviously, we have to cut off at 64, and that's essentially what we ordered and examined using the surface display. The one thing that is new here too is we extracted not that many, just 6 or 7 proteins that were previously analyzed. Probably would recommend using more, but we essentially found a ladder for stability, included that. And so that helps if you have different batches of proteases to adjust for how active and not active they are, because you just do a linear regression essentially. So yeah, then you sort the different concentrations. And again, this is from Gabe Rocklin, the original idea. And so here is an example of what we get. So essentially we had 30-something designs, 30,000 something designs, and a few random sequences that help you adjust overall to what's stable and what's not stable. I mean, finding the threshold. And then so essentially what I found for our batch here, anything above 0.5 was folded. So we have now over 7,000 designs that are folded and represent different folds, and here it's reflected where they all come from. And so you can use circular diagram to see, are they folded, how stable are they, and yeah, so that was pretty nice. So here's some general insights. We have also wanted to know if you have different backbones, if you redesign it multiple times, is it the backbone that determines the stability of the protein or not? And so the answer is absolutely not, because you can see a whole range of redesigned backbones, same 16 backbones that really span the whole spectrum. And I'll explain a little bit the motif. This is simple because we also discovered that you can use something that's been first described as a hash pair motif database by Jorge Fallas and George Rueda. The idea that you can extract pairs and use them for building interfaces for oligomers turns out pretty nice for designing course as well. And in fact, I can tell you that it significantly reduces the time you spend designing proteins, because first just look for the pairs that fit well, you minimize, and then they're actually so energetically favorable that you quickly find your minimum and don't spend much time redesigning the sequence much more. But stability bias, the only one where that made a significant difference was for 3 helical bundles, where you have essentially helices right next to each other, and it turns out that was where most of the motifs actually were extracted from. So depending on how you make your database, you can bias towards getting more stable folds. What you feed in is where it comes from. That's what it will do best, right? So mostly helical, and the more tight and helical interactions, the more the motifs improve the overall stability of the pool. So that's not specific. Okay, so then the things you can extract are, you can then figure out, now we have stable and not stable. You can figure out, what angles are possible to represent, what's the sheet dihedral, what is the overall curvatures, etc. And then you can look at like how one correlates with something else. But overall, I mean, there's quite some flexibility in the ferredoxin fold. As you can see, you have nice distributions. We were able to make a pretty tiny ferredoxin too, 50 something, which was very surprising. And then the other thing we looked a little bit more into was the ferredoxin fold, because nobody has designed a small ferredoxin. Nobody has designed a fold before, and this is a minimized version. So the significant part is, the parallel helices, and then the sheets going mostly one direction. This is one way of describing it. So we have ferredoxin, fluorodoxin, and so the synthetic ferredoxin would essentially have not these stabilizing extra helices. So, these are all the back in order to make sure these sheets form well. So it's essentially that connecting helix that you have to get rid of in order to make it fit into 64 amino acids. Overall, this motif is essentially what occurs in connecting domains. So this is pretty important. So that's an important motif. So sheet, beta, sheet. I mean, sheet, alpha strand is what is essential even in the Bossman folds. And so there, so that's actually a little spin. So there, that's D1, D2. There you have much more flexibility versus like if you look, for example, D3, D4, you have a pretty tight correlation between the distances there, not too much to change. And the NMR structure showed that our design looked like it was supposed to, at least the one we picked out. And so saying that, our algorithms probably work pretty well. Yeah, and so as I said before, we have now also more rules on how to connect helical bundles, and so when you have short connections, and when you expect them to be a small fold. And so we added that as well to the overall consensus designs that is available in Rosetta. And then of course we wanted to know, can we use this data set for machine learning? And the answer is yes. So we have a pretty decent area under the curve. So we did RUG curves here. So if you train on all and take out small subsets and do 5-fold cross-validation, you have pretty good power of prediction. So the AUC is really good. If you take, for example, subsets, of course, then you do pretty decent. This is classifiers. This is not, it's just a classifier, but that's good enough if you want to decide whether you want to by the design on that. Or the interesting part too is the dropout part is where you train on, you take out one fold and then train on all the other ones. Can you predict that fold that theoretically hasn't been seen before? And the answer is yes, you can use this chain model as well to identify stable, potentially stable folded proteins. And overall, I mean, what's not surprising is that it's the fragment quality that really drives this predictor. However, if you get rid of a lot of these other factors, the accuracy, of course, drops these values. All right, so I have to advertise this because we've been having problems publishing this because the reviewers are like, we don't believe that your proteins are folded because you don't really have much structural information. So the scaffolds have been used for a variety of applications. So here is like 12 designs, 12 targets that were targeted with this new algorithm using ref. Not going to explain that because not enough time, but that's essentially on the idea of like, massive docking of potential hotspots and then finding scaffolds to position on top of it. But essentially, over 80 or something % of the successful designs were coming from the scaffolds. So I hope the reviewers at some point believe us. We can publish all that as well. And the short story is the models have to be close to what they're supposed to be, which is also in the most recent paper, because if they're derived too much deviation, you can't redesign them. And so overall, they're doing pretty well. So I had to do that after dealing forever with reviewers' comments. So the other thing we're really excited about is this whole new software suite. A talented postdoc in my lab has been putting together over the past few years, which we call interface design, with 2 N's for neural networks. And so we have been working on how can we use deep learning for transient protein interactions and designing them. And so we made one assumption. We said, like, we want to focus on a small part because we can always transplant that into our scaffolds. And so the idea is, like, how do we make perfect contacts? The idea comes also from the fact that, protein interfaces have hotspots. So you do have to have only a few really good contacts to make sure you get binding, and then you have, some extras. But the idea is, how can we design something without explicitly just focusing on, like, making some disembodied hotspots and then finding scaffolds, present them. This should be all, if you train a network well enough, then it should be able to know these things without doing it explicitly. And so we have several algorithms that have multiple neural networks in them that I aimed to do that. And so the idea is we start with an anchor residue, and then we extract a small part of the binding site because we don't need the whole protein and it fits better into the arrays. So we started with PEP-BB to design the backbone. So again, this starts from picking an anchor residue, and then we interfacing with actually PyRosetta to make sure we're all biophysically we're doing the right thing. And then we start elongating from the initial peptide fragment, because we start with 6 mers. Again, this is idealized. It can be repeated multiple times. The overall architecture is the same as the PEP-BB part. And then once we have an elongated design, we can design the sequence using another set of neural networks. And so I wanted to start with introducing how the backbone is generated. So there we have 3 different models. One is for positioning, one is for predicting what secondary structure elements should be there, and then of course putting those 2 together. So first, you want to know what's the rough location, and then how should it look. And so that's encoded in an encoder-decoder system. We have 2 classification models and then regression model at the end. So the first one, again, so position, then what comes out of the position gets fed into the secondary structure prediction part, and then it puts these parts together. So we have curated the protein interface interaction training set and also test set. They were carefully curated based on what PISA defined as interactions, and then the proteins were relaxed and the pockets extracted. So essentially, we have slight variations between the sequence design algorithm and the backbone design algorithm. So one just takes 48 around the anchor residue, the other one is a bit more careful and looking into the spheres around it to make sure it gets the direct contact so it doesn't go too deep in the protein. And then here's a rough overview of the input we have. So we have, not surprisingly, distance maps and a secondary structure information that we keep, as well as amino acids that are used for training. And so for the sequence prediction, we also have the interdependence of within the peptide piece, the ligand part itself. And so one thing that's interesting too is, if you use N and O for the sequence prediction algorithm, actually work better than any other variations. And for the backbone, it's more on at least C alpha and then one other backbone atom. And so then we have also a descriptor of like how much something is buried or not. That was also important. Let's see. So then the way we look at this is for the positioning is, you have your anchor residue, and then you essentially put the coordinate system down there and see how, if you have a thinking about the 6-mer, if you look at position 2 and 5, because 1 and 6 can be dangling, which part of the coordinate system is close to these two positions, and that helps us to know the orientation on the binding site. And that's essentially a prediction. So it's a simple orientational description. However, occasionally you have cases where you have 2 and 5 potentially like equidistant to one of the coordinates, or one of the rays of the coordinate system, and then you don't know whether it's this way or the other way around. And as you can imagine, it's a lot easier to do that for helices than for other secondary structure elements. And so then the other challenge is, because we're not immediately testing with design and seeing whether it binds or not, we wanted to do a evaluation of native interactions. Do we see, can we recapitulate what native interactions do? But there again, you can be, depending on their interresidue, you have 2 residues that can be almost the same distance. So you kind of have to look a little bit shifted in order to figure out, in order to evaluate well enough the method. So like there's some challenges if you have a de novo design method, because there's A, multiple solutions to binding, and B, nature may not have the best one, but C, we want to know, does it at least cover it? So you have to give it a descriptor. And so what we did is, we compare a native 8-mers to 3 6-mers. So essentially, which would then cover the 8-mers, just simply because of the shifts that can happen. And so this is one of the benchmark results or test set results. So where the positioning, like the percentage of like having one of the 3 the right place in context of their orientation on the binding side. And so that's doing 55%. And as you expect, helices do better than, for example, let's say loops, or like just having it right on. Is a little bit lower, but it's still pretty high percentage. Similarly, predicting the secondary structure. Again, here we looked at, we start with already existing positioning, but it's like a secondary structure prediction in context of binding. And so that's pretty high and being able to do so. But then when you look at the final fragment position and backbone actual building there, we still have really close RMSDs to the native ligands. And so, well, RMSD4 is for the 4 core parts of the 6-mer, then the 6-mer here, and then part is interesting too, is like, if you just pay attention to what's hotspots, so there we actually get really close to the original interactions. So we were able to recapitulate that interaction, and that's super exciting. And so like here again, this is looking at the histogram, because the other one was of course averages. And so I think what's the most important is to get the hotspots right, because that's what carries the binding energy, and the rest can be mutated essentially quite a bit. So if your hotspots are positioned well, I think that means quite a bit. So here are some cool complex parts that just to illustrate what the RMSD actually mean when you would look at the binding site and then our cut out parts, right? Because we're looking at small parts and a lot of cut sites. So like for 5, if it's a helix, you're pretty close, and you can have almost identical contacts too. And then for sheets, of course, you have a little bit less flexibility, but still, this is how it would look. And it's pretty similar to the native interactions, which again, we're very excited about. And so here's an example for a protein interaction of an antibody and HA. So I've spent many years looking at HA interactions. So we are using that as our model system because there's so many examples and we know the contacts all really well. So let's say you start with a highly perturbed peptide, let's say something that would come out of PEPBB, so a predicted backbone. So if we use our sequence design, which is PEPZAP, and depending on PEPZAP 1 or 6, depends on how many outputs you ask for. You can see that we do a pretty decent job actually calculating this antibody loop. And so essentially what happens is that once you start having like a refinement step of the amino acid sequence design, it can help you refine the backbone a little bit more. And so I think that's also a big advantage over, like, say, just an energy function, because it actually helps you guide the backbone by knowing a little bit more about the sequence, which may be rejected if there were clashes really early on. And so fast design and comparison is not able to get the backbone similar or have any useful sequence to it in this context. So that was an example for the antibody antigen interaction. And so here's another one, also actually for HA, the head part this time. You have a loop that inserts in a pocket. And so here we are able to recapitulate the sialic acid mimicking aspartate in this CDR loop. And so that's pretty exciting. The same with the aromatics, or like having the hydrophobic contacts here. So that was all was pretty decent in this recovery set. And then the part that I find also interesting, as you think about converging solutions to binding, something Jim Wells discussed when first brought up the discussion about hotspot residues, is like, have certain kind of trends where, you have similar chemical contacts. So, you have a common chemical property. And so, you see that was how we started talking about hotspots, and they're clustered, have similar contacts because you have a certain chemical surface composition. And well, the shape complementarity, but what we started to see is that we also are really able to get these specific hotspot contacts. Similarly, as you see in those native interactions, so we sort of are having converging solutions coming through our networks. So these are some design examples. So yeah, we're pretty excited about this, and hopefully can test some soon. So then we wanted to know too about docking. How does it compare to docking, even though we are actually predicting the whole set, including the backbone. And so if you use the same kind of small ligand and then just do standard docking protocol, this is the Rosetta protocol, you actually very similar in numbers. So here's the RMC6, or like where top poses meaning best in score, and best poses meaning closest in structure. So here again, we have that too. We have the hotspots being very close. The overall RMSD6 or 4 is also very similar. Again, the hotspots is really what matters the most probably for design. And then when you look at the closest poses, yeah, you essentially comparable, even though you're starting from scratch. Again, that was pretty exciting. The other thing we were curious about is, if you were to take a complete entire surface of the protein, can you find binding sites? Like, do we see the privileged binding sites? Because these things have been trained on complexes, and the answer is yes, we can see preferred binding spots. So here is AHA again, and so we see several red areas where more motifs bound to. So we essentially have a rapid version of making a bunch of designs against the whole surface of HA. And so you can find areas where you get pretty decent contacts easily. And so then we look into where are the neutralizing antibodies located, and they essentially overlap with these areas, which we thought was pretty cool too. All right, so the sequence design, in the interest of time, I can't talk that much about anymore, but we compared, of course, native versus design overall, either trends, either matching, either recapturing what's there, and it does actually overall really well too. It definitely beats fast design. Yeah, here's another example of, when you have the backbone, does it recapitulate the antibody interactions? And so the answer is yes, he does a really good job with it. Yeah, he has just more examples for that. Contacts, it essentially has the same kind of contacts. It might not have the same amino acids, but overall, it has a rough idea of what's happening on the surface of these proteins. Anyway, all right, so I guess that was it. This is my group. Karen has done all the multi-state design work. Autumn has been doing a lot of work with the scaffolds. And Raulia has really written all of the neural network code and the benchmark and essentially did all that work. So she's did an amazing job here. And then the protein folds was together with Tom Linsky and David. RSV, Mark Tompkins here at UGA, and Jared Musa, and then Eric Procco gave me plasmids for ACE2 and whatever control. And yeah, that's it. Thanks.
Awesome talk, Eva. Questions?
I guess I can go. Great talk. I thought it was really cool that you were able to, even for some of the like design binder kind of hotspots that weren't quite recapitulating like the native binding mode, you were able to, after sequence design with PepSep, I think it's called, kind of like get closer to the native binding mode. And I was wondering whether you could talk a little bit as to how similar your starting point had to be to that final kind of like native binding mode to actually kind of get there. Like, is there a distance at which your sequence design method can't quite like converge?
Yeah, good question. 1 second. 1 second, the dog is barking like crazy. You can't really hear it. 1 second.
Oh yeah, I can't hear it.
Okay, so we haven't systematically looked at how far away or how much perturbed you have to be. But overall, they're pretty decently perturbed. I guess that's a number that could be looked into, yeah. But we haven't like systematically looked into the details of that.
Okay, yeah, it just seems kind of useful to be able to.
Yeah. No, I mean, how close do you have to be and have hope that you'll get there?
Okay, so I had another question, and this was related to the first section. So you were talking about stabilizing the pre-fusion conformation over the post. And I missed it, but I think you were looking at point mutations and like looking at the difference in predicted Rosetta energy in like one state versus the other, trying to maximize that. Okay, yeah, so I was wondering just, it seemed like you found a lot of mutations that worked. And I was wondering, it looked like if what they were doing was stabilizing the pre-fusion conformation mainly, but you could also imagine like destabilizing the post-fusion conformation. Did you find mutations that were like doing that as well, or was it mainly destabilization? Yeah,
so there are, yes, both cases exist, yes. The question is, I mean, but the original idea was to see whether you can, play with infectivity. Which we haven't done yet, and we're wondering whether we should be or not. But it would be interesting to know, like, what's the energetic difference, and does it affect infectivity of a virus, right? But what what people cared about was like, we want the pre-fusion, we don't care about anything else. So we're like, okay, I guess we'll make pre-fusion stabilization.
Yeah, okay, that makes sense. That depending on what you want to do, it really does matter to like balance the two states and how likely the protein is to be in either. I just thought that maybe it did seem like in the case where you didn't have this alternate state that you wanted to disfavor, it was kind of interesting to think maybe just stabilizing the state that you want is enough. Like if Rosetta can recognize what mutation, stabilize that, then maybe just you intrinsically get the benefit of destabilizing the other thing.
Yeah, no, I mean, this could be potentially just used without the second state. I don't think it's done that systematically this way. It might have. I mean, it could be that in some of Cyril Fleischmann's work that might actually use SSMs first, and then there's a combinatorial on that. I think it might, some of it might be in some of the PROS algorithms. I'll have to look. But I think, yeah, I mean, here it has an advantage by the post-fusion gives you the information where you're going to have good spots that are not optimized for that conformation. Right? So you just have additional information that you get through this. Versus like, if you wouldn't have that, you would be potentially less focused. But I mean,
That makes sense.
Yeah.
Cool, thank you.
Mm-hmm.
Eva, I had a question about maybe the first part of your talk when you talked about all the scaffolds that you generated. You mentioned that there's all these metrics that correlate to success of these scaffolds. Is that depend on the topic, or is that kind of does it depend on the target that it's being used for? You mentioned something about fragment quality and so on. Is that?
Oh, you mean like just for making scaffolds? So there is when you do your, you train, you have a classifier, and then you look, which parameters impact the predictive value capacities, right? And yeah, it changes on the fold. I mean, we mixed it because nobody in the end wants like a single classifier for one fold. Like everyone wants a mix that can be used for all, right? Like, I mean, I wouldn't want to like, filter all my designs based on fold and then have its own predictor. And you want a universal prediction. But so I did look into which parameters do change. And so what the short version is, fragments are very important the moment you have beta sheets, which you would expect. And the score actually is really good for helical parts, the Rosetta score itself.
Got it.
And I mean, yes, and there's a million other small parameters that will, push your AUC, above 0.8 and then 0.85. And so you just sort of inch up and you never know when you should stop. Right? That's the problem with like, predetermined features. So you can spend forever on it. So we found some other parameters that are predictive, but if you wanted to know what's the strongest one, I mean, yes, fragment score is the most predictive term. I mean, we looked also into like, highly connected residues need to have low energy, right? And that wasn't incorporated, and that's to some extent also predictive. So we added that. So it's beyond just the set originally reported by Rocklin, which is probably also, I think that the combination of having more folds, larger folds, as well as a few small extra ones, I think it's probably just a larger data set and then increments is a few other parameters. It's not very, what is it, the right word? It's not very satisfying finding these features because it's just itching.
Thanks.
Maybe following up, my question, the last point you mentioned about inching the accuracy up a little bit more. I mean, to get to a significant advancement in being able to predict the success of a de novo design, do you think that's going to come through just sort of like a steady march of small improvements, or do you think that there's going to be like a major leap at some point? Thinking about like an AlphaFold kind of.
Yeah. So I didn't show that, but yeah. So I already integrated AlphaFold too, and compared, and then you look at RMSDs, and then you actually do the whole score matrix over on just the models and then put all of these together. And there's another inch. And then the more parameters you then have on their AlphaFold prediction, there was another inching. And so it's like, I think it's going to be inching. I mean, unless you like write the perfect, network that does it. Right? But I mean, we were at success rates. Yeah, we haven't done an iterative process of like, now let me know those parameters, now redesign, and no, we haven't done that. I mean, you'll easily get over 30% success rate now, right? So, and then the moment you incorporate those, I mean, you can get pretty high in your success rates that it doesn't matter anymore. Like the moment you're over 50, you already have enough, right?
Yeah, totally. I mean, obviously the total dream someday is right as you synthesize, you design one molecule in silico, and you make it in whatever Craig Venter's bioprinter thing, and it comes out in a cure.
Yeah, no, totally. That'd be awesome. I think then we can, I don't know, we retire, yeah.
I remembered a second question I was going to ask, but just slowly coming back to me now. So in the second part of the talk, you talked about this transient interface design. I was wondering, how do you define like transient versus obligate? Is that separation that you did in the training? Or maybe I misunderstood
no,
that.
You know what, I didn't do a definition, but I mean, yeah, they're also separate, and they're predicted. So you can have a prediction that is trained on obligate and transient, but I don't think with these short fragments you'll be able to really make obligate interfaces. Yeah, I mean, we haven't looked into like big symmetric designs for this, mostly because we're interested in application that would, yeah, it could be something to look more into if you actually really set this up symmetrically. So the assumption is that this will result in transient, but again, we do have a trained system on obligate and transient, and we can compare that. But my guess is it's not going to matter.
Oh, I see. Yeah, the reason I was asking is because I think there's a lot of training from the PDB that's happening, and I think there's a lot of bias towards obligate stuff. And so I was just curious if there was a distinction that we were making or somehow biasing towards transient interactions.
Yeah.
Yes.
I mean, if the moment something starts working experimentally, we can give you all that data.
Nice.
Well, we soon screening a bunch of designs, so, and then if it works, I mean, let's see. So far, it looks very promising. I mean, the recapitulation and like the contacts look all really good. Yeah.
Oh, hey, I had one question. It's about the scaffold work where you were helping to characterize those nicely with those protease assays, those nice folds. I was wondering if you could comment on the integration of non-canonical amino acids to improve stability of these proteins. What if you're discounting folds just because they have one particular cleavage site, which could be kind of protected through some modification at that position? I guess it's a little bit outside the scope of the assay setup through the DNA synthesis, but would you say that this is it's certainly part of the capabilities, or is that to be able to model those types of non-canonical amino acids?
So you asking?
Well, I'm asking, I guess, if there are stable folds that integrate non-canonical amino acids. I guess there have already been examples of L&D hyperstable proteins, mini proteins. And yeah, is it true that this work looked at those types of folds?
We only do what you can encode genetically, right? Like you can't do high throughput.
Right.
Yeah. So I don't know how to do peptide synthesis, and so we just decided we'll do what yeast can make, and I'm not looking into any other ones. I mean, it's entirely possible. I mean, there's some nice work showing that you can like totally combine them in great ways, but then you're limited to certain numbers, and you might have more control over the shape instantly if you figure out, you know, how to put them together.
Thanks. Yeah, I guess it wasn't a really directed question, but it was just more your thoughts on that area of development.
Yeah, we won't look into it. We certainly will. Yeah, mostly because also we're very focused on interface interactions in the end. You know, the most recent study by Longxing Cao in Nature, you know, looked at 700,000 designs and had a success rate from 0.01 to maybe 1% for one target. So like, I mean, this shows it's, you know, it's possible, but it's incredibly hard problem. And so if you can't go high throughput at this point, it's going to be a little frustrating until it gets there.
Yeah, I mean, I guess you could dream about finding the most designable or stable like things that can display diversity in concentrated positions. Like, I guess you've seen some of those examples of non-antibody folds that are just diversified in a few loops.
Yeah.
To, you know, make a significant impact. So I'm thinking more about designing those types of proteins, like, yeah. What's that about?
Right, but I mean, designing a loop is also hard, right?
Designing a loop. Well, you demonstrated good. Didn't you demonstrate good loop recovery on the PEP-NN? Or
Yeah, no, we're pretty excited about that part too. We'll probably try them out in different fibronectins and camelides. Yeah.
Thank you.
Mm-hmm.
Awesome. So if there's no more questions for Eva, let's thank her one more time for an awesome seminar and great discussion.
Thank you. Thanks, everyone.